The Neurotransmitter Epinephrine by Audrey and Angelina.
The chemical structure of epinephrine includes a catechol group, an ethylamine side chain, and a methyl group. The catechol group, which is a benzene ring with two hydroxyl groups, makes up the core of the molecule. The hydroxyl groups, which are adjacent, are attached to this ring and are attached to the first carbon of the ethyl bridge, which is made up of the ethyl group, and is attached to the third hydroxyl group. The amine group is attached to the second carbon of the ethyl bridge. Lastly, the methyl group is attached to the nitrogen atom.
Produced in the adrenal medulla, epinephrine is synthesized through a series of enzymatic reactions. The hydroxylation of tyrosine, which is an amino acid, takes place in the chromaffin cells of the adrenal medulla, where tyrosine is converted to the levodopa by the enzyme tyrosine hydroxylase, and then converted into dopamine by dehydroxylase. Next, dopamine is converted to norepinephrine by the enzyme dopamine beta hydroxylase and then transported into storage vesicles via the vesicular monoamine transporter. Lastly, norepinephrine moves to the cytoplasm where it is methylated into epinephrine by the enzyme PNMT.
After being synthesized, epinephrine is in the cytoplasm, where it is then transported into storage organelles called chromaffin granules by using vesicular monoamine transporters. Epinephrine is stored and protected from degradation. The high concentration and protection ensures for a readily available rapid release for flight or fight situations. After its release, epinephrine is initially converted into metanephrine by catechol methyltransferase. Next, monoamine oxidase removes the amino group from metanephrine, which results in aldehyde. Aldehyde is then oxidized into vinylmandelic acid, which the kidneys excrete. Unlike other neurotransmitters such as norepinephrine and serotonin, epinephrine is not recycled back into the nerve terminal by a reuptake mechanism. This is because epinephrine is near complete degradation by COMT and MOA.
Epinephrine is found mostly in the bloodstream but also in the brain. Because epinephrine is not neuron specific, it is not limited to synaptic communication and thus can act systematically. Its lack of specificity is attributed to the fact that it is produced by chromaffin cells, which are non-neuronal cells. The neurotransmitter's flexibility allows for a dual role. Epinephrine can act as a neurotransmitter and a hormone. Because it is not confined to neural circuits, epinephrine circulates the blood and reaches many tissues, leading to widespread effects including the body's fight-or-flight response.
The 9 adrenoreceptors, which are a class of G protein receptors, can be divided into two categories, alpha and beta. These receptors, while active, induce vasoconstriction, inhibition of the release of norepinephrine, and stimulation of cardiac output. Binding to alpha-1 receptors, which activate the GQ signaling pathway, triggers intracellular calcium release. Binding to alpha-2 receptors induces cellular activity inhibition as it couples with the GI protein, leading to the inhibition of cyclic AMP production and opening of the potassium channels. Binding to beta receptors, which primarily couples to the GS protein, activates adenylate cyclase and increases cyclic AMP levels.
Epinephrine causes a depolarization mediated by the beta adrenergic receptors, which is associated with the decrease in the membrane potential, thus suggesting the closing of potassium ion channels. This depolarization is driven by the activation of the G protein pathway, which leads to the suppression of voltage-dependent potassium ion channels. This inhibition of the ion channels makes the postsynaptic cell more excitable and available for firing and action potential. Activation of the alpha-1 adrenergic receptors also modulate synaptic transmission by increasing the frequency of excitatory postsynaptic currents. Alpha-2 adrenergic receptors act as autoreceptors to inhibit epinephrine release.
By activating the adrenergic receptors, epinephrine influences functions like food and water intake, respiration, blood pressure regulation, and thermoregulation during a flight or fight response. This response starts with triggering the brainstem, which is necessary for epinephrine to send signals as it does not cross the blood-brain barrier. Starting in the brainstem, epinephrine activates the beta adrenergic receptors on the vagus nerve's sensory afferents. Epinephrine also uses a signaling pathway to promote an enhanced state of alertness.
Epinephrine malfunctions can produce a series of diseases. In dopamine beta hydroxylase deficiency, the conversion from dopamine to norepinephrine and epinephrine is prevented, resulting in undetectable levels of catecholamines. Neuroblastoma is a cancer that affects the adrenal glands, leading to low epinephrine. Paraganglioma is a tumor of the chromaffin cells, causing overproduction of epinephrine. Also, extreme stress can cause broken heart syndrome, causing a surge in epinephrine production. Mutations also lead to hypertension, arrhythmia, and genetic syndromes. The excessive epinephrine production can also lead to cell death, leading to cardiovascular damage and tumors.
Alpha blocker and beta blockers both block epinephrine receptors. Alpha blockers specifically block adrenergic receptors to prevent epinephrine from binding and activating target cells, thus reducing its effects. These blockers decrease blood pressure by preventing vasoconstriction and are used to treat hypertension. Monoamine oxidase inhibitors and tricyclic antidepressants are antidepressants that prevent the uptake of monoamines and lead to increased levels of epinephrine in the synapse. Natural toxins such as trace amines also produce vasoconstrictive effects by directly interacting with adrenergic receptors. Ephedra and cauterides are natural stimulants that can mimic epinephrine effects. Lastly, 6-hydroxydopamine can metabolize into toxic metabolites to damage epinephrine.